In this final segment on biomarkers, I want to present a framework for thinking about biomarkers and biomarker development, realizing that the where a particular biomarker seems to be useful in this scheme frequently breaks down, and it's always valuable when that happens, when a biomarker transgresses our boundaries. Now, first of all, let's think about whether a biomarker might be useful in pre-therapy versus during therapy. Now, first, again, and then let's think about them in terms of the characterization of the disease, which I want to distinguish from saying that there's no consideration of the therapy at this phase, but simply put, we're placing the utility of this biomarkers in this category as happening when we're deciding what to do about the disease. So there are prognostic and predictive biomarkers, and the distinction between these might be fairly fine, but in a prognostic indicator, we're really thinking about the outcomes. And in a predictive biomarker, we're talking about this might be in terms of stratification of treatment. And I'll talk about a little bit about some examples here, but basically, how aggressive might the cancer be, versus whether the patient has a particular duplication or mutation that might make them a particularly good candidate for some treatment down the line. And then, of course, there's going to be risk assessment biomarkers. These might be valuable in terms of deciding, in terms of determining whether a patient is at risk for a particular disorder or is at risk for a particular toxicology. So there's disorder versus toxicity in terms of risk assessment. Now, turning for a moment to thinking about what happens during therapy, we're going to consider a utility of a biomarker in terms of optimizing a dosage regimen for a particular patient. And also, we might think about using the biomarker as a surrogate endpoint. That is to say, what's a surrogate endpoint? Well, usually we're talking about when we're talking about an endpoint, we're talking about the outcome, right? And that might be the survival of the patient, or the rate of metastasis in a patient when we're talking about cancer, as we have been for most of this talk. But now we're looking for a correlation between the biomarker and the outcome. And if it's robust enough, then perhaps we can use the biomarker itself as a surrogate for the real endpoint. We don't have to wait for the outcome of the disease. We can make some judgments based on just the presence or absence of the biomarker. So again, these are just some useful categories, just to give you a sense of perhaps some of the examples I'm going to talk about now. Prognosis, something that's common to use, that's not a pharmacogenetic biomarker, but still it's one that I'm going to talk about as a preliminary example, is fluoroglucose avidity. So again, the aggressiveness of a tumor, the prognosis for the patient as measured by FDG avidity. Circulating tumor cells is another one that we're going to see examples of in a few minutes. And then, of course, we can use HER2 status as a prognosis for some, as a prognosticator or a prognostic biomarker in some kinds of cancer. Particularly useful as a predictor, which another one we've already talked about, is KRAS status, as a predictor of whether a patient, regardless of their prognosis, whether a patient will respond to a particular kind of therapy. And of course, during therapy, we can use HER2 status to judge whether or not a patient will respond to a particular drug. Of course, I'm going to show you an example in a minute of using, again, fluoroglucose avidity of a tumor as a way of determining a dosing regimen. And so not only, and then again too, we'll use circulating tumor cells to see how as a surrogate for actual outcomes. Um, Again, the point being that these biomarkers cross boundaries regularly, um, and that's valuable, right? Obviously, it's of use to have a biomarker that we can use in a range of different kinds of setting, but it's again still useful to make those distinctions as well. Right. So where that biomarker falls in that list of possible utilization comes from how you use the biomarker, how you select as the physician or investigator to use a particular biomarker. And here I'm starting out with an example that has nothing to do with pharmacogenetics, at least not blatantly or directly, but it gives us a sense of what other kinds of biomarkers are out there or have been in use up till now. I want to hasten to add that um, this slide is about imaging, and as your Kelloff review points out, no drugs have been approved using imaging-based biomarkers as the driver. So I want to emphasize that. However, there's lots of interest in using imaging based biomarkers uh, in a variety of contexts. And so this is something that is coming and happening, development happening in parallel. And again, this is technology driven. Prevalence and cheapness of technology and ease of the application of the technology, very important, as we referred to earlier on in these lectures, in terms of the development and in the utility of a particular biomarker. So how easy is it to image the patient regularly, frequently, versus how easy and cheap is it to perform the sort of sequencing tricks that we're going to talk about in the next couple of slides. In any case, this I'm referring to um, F, fluoro-D-glucose as an imaging agent. It's a positron emitter, so we can use it as an indicator of metabolic rate in particular kinds of imaging technologies. This is positron tomography. But what I really want to point out here is the utilization of this as a biomarker in a prognostic or diagnostic setting as well as in a dosage setting. So again, we're using these indicators in different contexts. Here we're comparing the PET image with a CT scan, and what I want to point out to you, maybe let me try some color changes. What I want to point out here is the tumor. We're looking for tumor metastasis, and so we see the spread of this tumor throughout different tissues, and we seem to see similar behavior in both technologies. Now, this is useful just in terms of validation of an imaging a cross validation of an imaging technology. In any case, we can use this FDG avidity to find the metastases in this particular kind of cancer, and I don't even care about what kind of cancer we're talking about in this case. So bear with me on that score. And then again too, so we can make a prognosis about whether a particular cancer is in metastasis or is about to metastasize. Um, and then too, that we can talk about the relative utility of particular imaging technologies, in which in this case, we can see FDG uptake. This is in the pulmonary. Oh no, sorry. This is in rib metastases. We see them in the FDG avidity survey, where we miss them in the more conventional CT scan. So prognosis versus dosing regimen, and so in this particular slide, come back to my pin color. We're looking at this is actually, to be fair, is a convolution of the FDG imaging with tumor size. So this is basically some kind of ratio, or I'm going to say that we've convoluted FDG imaging and tumor size or tumor density without belaboring the details, I just want to point out to you um, how we can use this kind of biomarker as a tool for adjusting a therapy. And basically, here what we're seeing, what I want to draw your attention to is that we have four different dosage regimes here represented in different colors, and we're making a lot of this, but basically and here's the chemotherapy agent. What we see is that there's a difference between the low and high the low and high dosage regimes that's apparent even after a couple of days of chemotherapy. So this is a useful tool, the authors here are arguing, as a way of adjusting the dosage of a particular drug based on something that's primarily driven by um, fluoroglucose imaging technologies. So again, the point is that we've got this structure for thinking about what kinds of biomarker a particular test might be, and we talk about crossing those boundaries all the time. Here's one more slide of imaging markers, not really a cytogenetic marker, but in this case, the imaging, and this is largely, well, this is some varied imaging methodologies all rolled into one here. But the reason I'm showing you this slide is again because Keloff, in his review that I assigned you, refers to a variety of biomarker types that are in development in addition to sort of straight up pharmacogenetic or pharmacogenomic markers, and these imaging markers are some up and coming ones that are worth talking about. Again, they illustrate some of the same things as we see with our pharmacogenetic markers as well. In this case, the imaging technologies, which are varied here, basically are telling on the molecular phenotype of a particular kind of circulating tumor cell. So here in particular is what Keloff is discussing, circulating tumor cells. This is just an image that I stole from Scripps, and it doesn't even matter what kind of cancer this is. What I'm trying to show you, illustrate to you here, is the kinds of things that we can observe in this kind of with this kind of imaging technology. This is just various kinds of fluorescence imaging sort of all rolled into one, including antibody-based markers, as well as, here's DOPI, which is just another of the intercalating dyes. We've discussed intercalators in other cases, so this is a nuclear marker here. Now, in this case, we're talking about cytokeratin, which is a marker for lots of carcinomas. Again, I'm not going to be specific in this particular discussion about exactly what kind of cell we're talking about, but suffice it to say that with this kind of imaging technology, we can often define tumor phenotypes, and additionally, we can look into the prognosis for a particular patient, given a variety of cancers. These are in development for breast cancer, prostate cancer, various others, lung cancer, several others, and nothing definitive as yet, nothing clinically useful perhaps as yet. But the point being that we can develop these kinds of imaging techniques as prognostic biomarkers. I was also going to mention CD45. This is a cluster of differentiation 45. This is another antibody-based stain, and we see that the tumor cell is actually CD45 minus in this case, and I'm not going to talk about why, but suffice it to say that the expression of CD45 at the surface of tumor cells is very highly variable. In some cases, the tumor cell overexpressing, in other cases underexpressing various versions of CD45. The point being that we can draw some detailed conclusions about this molecular phenotype of the tumor based on this sort of differential fluorescence imaging. And I think that's all I need to say about that. But the bottom line is too that these can serve, as Keloff mentions, in a variety of contexts, not only defining phenotypes and prognosis of the tumor of the patient, also looking for surrogate endpoints as predictors, right, of the outcome of the disorder, as well as the usual suspects apply here, as well as looking at drug efficacy and response. Now, again, the theme that I keep hitting on in all of these slides is that we see, once we've decided on all of these terminology to apply when describing um, molecular biomarkers, that we often uh, apply in a variety of categories. So in this case, I've just taken a couple of pretty famous papers on HER2 expression and cancer prognosis and drug response. So here again, too, we've got multiple ways of utilizing the biomarker. In the first case on the left, this is a paper, from 1987. We see that the point here being that HER2 new copy number correlates with low survival rates overall. So that's the overall outcome, and it's really clear here. If we look at let's just focus on the top case, for example. We're looking at survival. We see this difference. Right? Without amplification of HER2, the prognosis is very much better than with that high copy number of HER2. But not only that, we can also see the use of this in the drug, evaluating the drug response to Herceptin, which we've mentioned previously. Mentioned previously in this lecture. And so also we can look at, when we're talking about the enriching. So in this case, the study has been enriched, a term that I've used a few times today. Enriched for HER2 overexpression, we see that these patients have a clear difference when treated with trastuzumab, Herceptin, versus those that are treated with standard chemotherapies alone. So pretty significant differences there. So again, the point simply being that the single biomarker can overlap in a couple of different ways at different stages and different stages in the treatment, both in the terms of the prognosis before treatment begins and then as the treatment proceeds, as I've mentioned several times. Now, I'm going to finish this set of lectures with a couple of examples, and I'm stealing figures from a couple of the papers that I gave you to read this week. In this case, we're looking at, finally, pharmacogenomic markers and the evaluation of AML tumors. This is from the Patel article. This is a really fun article, I think. These plots are sort of getting famous now. These are made with Circos, which is a great program for presenting tabular data in a circular format. The upshot here is that we're looking for a genomic prognosticator. I like phrasing things that way. Basically, the problem in this kind of tumor is that there are very heterogeneous presentations of the tumor, and it makes it very difficult to clearly establish exactly what tumor type and exactly what prognosis the patient might have. And so we're looking for a way to differentiate between patients. And in this case, the authors look at a whole slew of genetic alterations, and what they observe for a very large number of patients, this is around 400 patients involved in this study, they look at this 18 gene panel, and I won't describe all of these right now, but suffice it to say that all of these patients, nearly all of the patients, show at least one and usually more somatic mutations in the panel that they selected to study. Some of those are simple mutations. Other of those are very large duplications or translocations. So here's a duplication event, just to point one out. Some of these we've seen before, receptor tyrosine kinases. Some of these here's KRAS as an example of a downstream signal transduction molecule, and here's again Here's isocitrate dehydrogenase, another metabolic player. It doesn't really bear on our discussion today to look exactly at what the molecular basis of all of these alterations are. But the point being, in this slide, they're looking at the frequency with which these patients show um, a multiple substitutions within the same tumor cell. Um, and so the upshot here is that if we're looking now, we're focusing on a particular gene, DNMT3A, and in this case we see that very frequently is this substitution or this modification or this mutation found associated with NPM1 as well as with FLT3. Right? And then there's others as well that are less common, but for sure we see a very clear signal standing out here that these substitutions, this set of 4 substitutions, all co-localize. And we see something similar in this side where we're focusing on FLT3 and how it compares, and we see that the reciprocal case is true. Obviously, in this case, FLT3 is associated with DNMT3A substitutions as well as with NPM3. So the point is that we see a signature of mutations emerging that we can use to define the prognosis of the patient, and maybe as well as we've seen in other cases, classify the tumor much more carefully. So in this case, we'll see that not only does the genome type predict survival, which I believe I'll show you on another slide, it also can affect treatment outcomes and help determine treatment types. Right, here they are. And so a combination of known cancer-related genotypes. So what it boils down to is that, at least for a large number of the members of that panel that I described of 18 genes, many of these are sort of usual suspects in terms of what kind of role they might play in the tumor. So here's a receptor tyrosine kinase. NPM3 is involved in tumor suppression. Here's a transcription factor. TET2 is a DNA modification. Here's obviously a DNA methyltransferase. So again, DNA modification. IDH2 I mentioned before is that metabolic marker. That's again accumulation of toxins, which we won't describe in any great detail here. But the bottom line is this again. I like this kind of phrase. This is usual suspects used in this study to try to identify a molecular genotype or molecular phenotype for what sorts of, if you want to think of it that way, what sort of substitutions are present in a particular tumor type. And they assign then a risk profile to particular sets of linked mutations. And that's, I'm not going to describe in detail what those sets of substitutions were, but we can see that we're able to stratify the prognosis or the eventual outcome based on the genotype. So in this case, they have a so-called intermediate cytogenetic risk, very much more long-lived, very much more long-lived than those with what they deem to be the unfavorable cytogenetic risk. So by looking at the genotype, correlating the usual suspects with outcomes allows them to develop a biomarker for, in this case, outcomes, right? Survival, prognosis, but also with treatment style. So that now, again, I'm glossing over a lot of the details here to try to give you a sense of overall of where this work takes us. In this case, they can look at not necessarily a complicated genotype, but 3 substitutions or 2 substitutions and or a translocation event, and they can say that the presence of any one of these allows us to segregate between treatment styles. So here's daunorubicin. We won't talk about the molecular basis of daunorubicin, and again, in this case, I think we've covered that in previous lectures. But the bottom line is that we can tell, according to what substitution type, how a particular dosage is going to serve that patient. So it's very clear here that we see a major difference in the overall survival of this category of patients depending on the dosage of the drug. So, again, this is an example of an emerging and potentially extremely robust means of making a prognosis and devising a treatment strategy based on a genomic assignment strategy. In this final example of biomarker development, which I'm calling a discovery example, I hope that you will agree with me that I haven't diverged too far from our discussion of biomarkers. So this is very much an emerging story, and nowhere in this article by Matsumura from last year is the word biomarker even mentioned. But what I do want to suggest to you is that even though this is a very new line of research, it even so is still pointing to potential therapeutic strategies, which I'll come back to at the end. Now, this is an example of an epigenetic study, and it's a complicated study, but I think that one thing that's interesting to me in this kind of study, which is we've seen several times before, is that the research begins with a careful examination of the literature for development of a potential list of potential candidates for that the workers devise a hypothesis for or base that hypothesis around. In this case, we're looking at ovarian cancer, and the workers are basing their hypothesis on some clear observations about gene silencing, so methylation leading to silencing of a variety of genes, in particular TGF beta, transforming growth factor beta, and and as well, and ultimately then the deregulation of that pathway, which is observed in lots of cancers as sort of the molecular basis of that tumorigenesis. Now, in this study, then, they're looking at methylation of a variety of genes, and when they begin with this panel of 300 some potential genes, they observe that these tend to be enriched, and this is building on previous work of theirs, but these tend to be enriched for TGF beta pathway members. And they developed, and again I'm glossing here, right? But they developed some pretty stringent inclusion criteria, leading them to monitor carefully 50 of these candidates by methylation sensitive or methylation specific PCR, followed by some pyrosequencing, which those are methylation sensitive PCR we haven't talked about. Pyrosequencing we have. I won't talk about that. But in any case, they're monitoring methylation of these genes and trying to correlate that with expression patterns. So, and there's the trick, right? We all know that epigenetics is all about how methylation can alter transcriptional regulation but now in this case they're looking specifically for relationships between particular genes and expression patterns within a particular tumor type. I have a couple of data items to show you here that I want to more sort of carefully, even though carefully yet tersely explain. In this case, we're looking at cell lines, cancer cell lines. I'm not going to talk about which ones they use. I think ultimately it was some 30 odd cell lines that they utilized. Let's suffice it to say that they were exhaustive. And there are two data sets presented here. This happens to be for one gene, which they did a whole slew of genes, but we're only looking at one, for transforming growth factor beta receptor 2, and they correlate expression, which is here in this sort of heat map diagram, and we see low expression here in green and high expression here. And I'm reminding you that these are in different kinds of cell lines, but what they're also correlating at this point is that expression pattern with hypermethylation. And so that's what this part is hypermethylation. And this is an average over actually um, several genes that they were analyzing in each cell line. But suffice it to say that what we see is a strong correlation between expression patterns and methylation, which is entirely consistent with what we expect. The point is that they're using that as a basis for looking for correlations again between methylation and expression and those expression patterns in a tumor specific way. And so that's what this bottom uh, figure is showing us here. They looked over some very large number of tumors, 146 patients, and used unsupervised hierarchical clustering. These are terms that you're familiar with now from reading all of these microarray papers. And they see that the tumors can be broken down into two groups. Oops, there's group 1, two groups based on methylation patterns, and there's group 2. And I think you can clearly see that in group 2, well, here's the clustering. There's that, and there's one cluster and another. And we can clearly see that there's differences in expression of their gene set in this second group, and it's less clear that there are really big changes or patterns in expression in that first group. But the key distinction between these two is age. So we see age-dependent silencing. So in group 1, these are young women, and they see TGF, plenty of TGF beta activity. But in group 2, these are aging patients, and they see hypermethylation in this panel of genes. Now, so this is where I'm emphasizing the fact that this paper fits into our discussion of the development of biomarkers. Again, early days in the development of this as a biomarker, but this ability to stratify the patient population according to the correlation between methylation and expression can lead to these therapeutic strategies that I mentioned. So in the case of group 1, potentially a therapeutic strategy would be TGF beta inhibitors, which exist. I'm not going to discuss these as inhibitors, but beyond our discussion today. But as well, there's a potential treatment pathway for the second group of patients, from which there's a strong and obvious signal of methylation silencing of the TGF beta pathway, and that would be DNA methyltransferase inhibitors. So, which obviously are all about hypomethylation. So here again, the point being that we're using very novel technologies to, as well as in combination with fairly standard technologies, to try to find a signal that allows us to segregate the patient population, and that ultimately allows us, when there's nothing here about prognosis, right, which of these patients is going to survive longer than the other. That's not the point. But in this case, we're talking about jumping ahead to potential therapeutic strategies. Now, let me hasten to say as well that coming at the end of the paper, as it does, that sort of hand waving or speculative argument is not strong, but it's still significant enough, I think, that we can look at this as an emerging example of biomarker development. I think the only other slide to show in this case is this methylation sensitive PCR. This fits in with our methods discussion of previous weeks, and this is simply basically a PCR method with an additional step which uses bisulfite as a DNA modification agent, which will convert unmethylated cytosines to uracils ultimately. The key, of course, being that methylated cytosine is not a substrate for for the sulfonate formation. So when we have a genomic DNA that bears a methyl group at this cytosine, and we treat it with bisulfite, those unmethylated cytosines are converted to uracils. This is maintained as a methyl cytosine, but the methyl cytosine is still complementary to its G, where obviously the U's are complementary to A's, and so under amplification, we retain as a C the methylated cytosine, and then we can go and observe that by standard sequencing methods. So that's the basis of that methylation specific PCR. I thought it was worth throwing that out there as yet another trick in the tool bag for identification of potentially interesting pharmacogenetic phenomena.